Thank you. Thank you for inviting me to present Vecura. So we are a Karolinska Cell Therapy Center, and we also, I will try to remember to keep this one over here. You can hear me. We're also part of the Karolinska ATMP Center. So we are a manufacturer of ATMP medicinal products. Yeah. Okay. There. So I will shortly just introduce what ATMP is for those of you that don't know that yet. I see many faces that I recognize here in the audience, so I think many of you already know this, but it stands for Advanced Therapy Medicinal Products. And it's a quite new type of medicines that are coming the latest 10-20 years. And they are usually they are divided in three groups. You have gene therapy products, that is, you transfer DNA or RNA to the patient or you can also have cells that you gene manipulate before you deliver it to the patient. You have cell therapy products that contains cells or tissues. Yes. And you have tissue engineered products to cells or tissues that can repair or restore some tissues in the patients. And it's the Medical Product Agency that is the authority in Sweden that regulates this type of medicines. We are a core facility at the Karolinska University Hospital, and we produce ATMP products mainly for clinical trials, for phase 1 and 2 clinical trials. And we have authorizations from the Swedish MPA, so it's a manufacturing authorization, import from third countries, wholesale permit, and also tissue establishment for procurement of the cells and tissues that we use as starting material in our products. And our mission is to facilitate the development of ATMP for the treatment of diseases that currently lack efficient treatment, and support the translation from research bench to the patient treatment. So all the processes needs to comply with good manufacturing practice and all the medicinal laws that regulates these kind of products. And we are working a lot with Karolinska Institute and the physicians at the Karolinska University Hospital, as we are a core facility, but we also work with other academic institutions in the country and also with customers outside, smaller biotech companies and sometimes bigger companies also. So we are open for that. As I told you, we belong to the ATMP Center, and so that was Karolinska Institute and the hospital that want to gather all the ATMP activities, both from research and process development, that is mainly performed at the KI part, and the GMP manufacturing to be able to treat the patient, and that is more under the Karolinska University Hospital. A short history of Vecura. So even though that maybe you have heard our name, you have seen our people here, but we have been here for almost 30 years now. So we are situated in the floor 3 and also the floor 4 of the G part of this house. So we had the inauguration in 1996, and in 1998 we did the first Swedish GMP manufactured gene therapy product in a clinical trial for cardiovascular disease. Later on we implemented manufacturing of cell therapy, and in 2008 we did the first manufacturing of cell therapy product for Alzheimer's disease. We have also produced hospital exemption products, and we started with gene therapy manipulated cells in 2014 in collaboration with Uppsala University. So we were the first European GMP manufactured CAR T cells. And then we have the ATMP Center that was established and inaugurated this fall in August 2025. We have a long experience in GMP manufacturing, over 25 years now. So we have been working a lot with plasmid DNA also, and cell therapy products. We have been working with many different kind of cells, CAR T cells, immunology cells like NK cells, TILs, epithelial cell sheets, and mesenchymal cells, and also some pluripotent cells. And by that we also have worked with different culture systems and also in different scales, both in like small culturing flask and also bioreactors, but they are usually kind of small bioreactors. We do also do some import of viral vectors from the United States. So ATMP products must be sterile since they are parenteral products, but they cannot be terminally sterilized since they are living material we are working with. So you need to have rooms and a facility with very high cleanliness grade to maintain a sterile or aseptic environment. And also you need a high grade of the gowning. So I try to include a lot of pictures here to you can see how it looks when we are working in these rooms with the manufacturing. And you need to have good workflows designed to ensure the quality and safety of the products. So these premises, they are built as you have a good design and layout, and surfaces and materials needs to be easy to clean. All the air that comes into the rooms needs to be HEPA filtered, and you have differential pressures between the different rooms to allow a good airflow from the higher pressure in the cleaner room. It goes so you get the right airflow in the facility, and airlocks between the different cleanliness grades, and you need to monitor it, and you need to perform a lot of environmental monitoring to show that you have good levels of particles and microbes in the facility. This is just to show shortly how we are. We are situated, the most part is in floor 3. So it's just below over there. Maybe you have seen when you have walked outside the Novum. You can look in, and sometimes you maybe have chance to see us in the green suits walking around and wondering what we are doing there. So here is, I think, over here is the entrance of the Novum from the Hälsovägen 7. So we have 10 grade B clean rooms. That is the highest level of cleanliness, and that is in the rooms that we are producing. Then we have several other rooms connected to these production rooms. We also have one clean room on floor 4. So to produce ATMP products are challenging in many ways. And most of these are produced as one batch to one patient. They are often autologous products, and that means that you take the cells from the patient and you produce a cell product that is then returned to the patient. And our flow is that maybe a patient is asked to participate in the clinical study. Then we receive an order from the physician. And then manufacturing often needs to start within 1-2 weeks. So we don't have that much heads up. And cells are collected from the patient, or if it's a donor, and manufacturing process take about 1 to 2 weeks to perform. And some products are what we call fresh products, and they are given to the patient almost directly on the same day they are ready. But there are some also that are frozen and can be delivered later on. So when we have several ongoing projects with different conditions and different demands, it's very difficult in planning. And so everything that happens in this building with power failure or ventilation problems, then it will affect our production. And it's very difficult to sometimes plan and be long ahead of what is going on. I will also tell you a little bit about 4 of the ongoing current processes. So this product is called PTTL, Personalized Tumor Treated Lymphocytes. And it's a clinical trial that's ongoing. It's a collaboration with a biotech company called NeoGap Therapeutics, situated in Solna. So here is a treatment of patients with colorectal cancer, stage 4. It's a late stage of the disease. It's an autologous product. So I'm just going to try to explain this. This is very personalized product. You try to train the T cells, the immune system, to educate them of the patient's own tumor. So what you do is you take out a biopsy from the tumor and you sequence it, the DNA and RNA, and you compare that with a healthy tissue and try to find the epitopes that are for the cancer. So you can, the company has a platform, a software called Pure that can find these specific epitopes. And then you can produce a neoantigen, and you couple those on magnetic beads. This is a critical raw material that is produced by the NeoGap. In the meantime, we take the lymph nodes that are tumor drained, containing hopefully a lot of T cells from the immune system that are activated already on the tumor, and also containing antigen presenting cells. So this mixture of cells, you co-cultivate them with these neoantigen coated beads. And the APC cells, they pick up the neoantigen and present them for the T cells. So the T cells get educated on your own specific tumor. I hope you. And then you have this product, and you take away the magnetic beads, and you treat the patient. And another product called CAR20NAPT. Maybe you've heard of Göran, that has been in the newspapers and on the morning studio TV4. I think it was just a couple of weeks ago. And Göran was the first patient in this clinical trial that received this product. And it's a treatment of patient that has cancer, blood cancer, B-cells lymphoma. And it's a collaboration with Uppsala University and Akademiska sjukhuset. And it's an autologous product. And in this case, this is a gene a gene-modified T cells product. And here we introduce genes to the patient's T cells. And the gene you use a retroviral vector that contain these genes of the CAR20 NAP. And this one will then be introduced to the T cells, and it will be translated and presented on the T cells cell surface. And the CAR20 will find your B cells, that is the cancer cells, and kill them. So what we do is that the patient comes to Karolinska sjukhuset in Uppsala, and you do a blood harvest with leukapheresis and a cell separation. So we get the PBMCs, and they are transferred to Vecura. Then we load them on a bioreactor called Prodigy, and then we do the transfection with retrovirus. And then we expand them in about 8 days, and then we formulate them, and it's a frozen product, so it's shipped back to Karolinska. And then we have another interesting project called donor-specific immunomodulatory cells. And it's also a clinical trial ongoing, but we have not treated any patients yet, but we are waiting. And this is treatment of patients who have undergone liver transplantation, and that is to reduce, they are on heavily medication for immunosuppressive medicines through the whole life. And it's very tough treatment. So this product will aim to reduce this medication, so it prevents the immune reaction against the liver. It's also autologous, but here you use, you have the deceased donor that will donate the liver to the patient, the recipient. And on the same time almost, both the donor and the recipient will do a blood harvest with leukapheresis. And after that, the recipient receives the liver as a donation. At the same time, the PBMCs from the deceased donor, they are irradiated so they cannot proliferate, but they are still alive, right? And you co-cultivate those together with the patient's PBMCs. And then you use what do you call it, antibody that so your cells do not react on the donor cells, and you co-culture them for 2 weeks. So here you train them to tolerate the donor cells. And this is a collaboration with the transplantation unit here in the hospital in Huddinge. And the last one I also want to show you, it's a clinical trial that will start next year in the spring. It's a collaboration with KI and the Allogen Stem Cell Transplantation Unit and ATMP Center in Oslo. Here, treatment of patients with refractory high-risk myelodysplastic syndrome and acute myeloid leukemia. And this is a little bit, in one way, complicated how it works, but let's see if I can. It doesn't work. Here you have selected donors that have a specific kind of NK cells that, so we call them super donors. They have a lot of single, like KIR, it's a special molecule on the NK cells. So you use usually NK cells recognize what is wrong, that they recognize your your set of cells in the body. But if you are missing your own, then they react. So here you try to mismatch from the donor to the patient. And to be able to expand these cells, you need to have feeder cell banks, feeder cells that express HLA-E. So we need to have one feeder cell bank that also is irradiated so they cannot proliferate in culture. And you have this super donor cell bank that you co-cultivate to be able to expand them. So it's called adapt NK cells. And I just want to show the Vecura team here. We're not all of us here. We have expanded the latest half year here, so we are almost around 20 people now. But here, some of us, maybe you will recognize them here in Novum when you're passing by. Thank you.